People are always interested in health, anti-aging, or longevity. How long different species live depends on several important biological processes, signaling mechanisms. These processes include ERK, STK11, also called LKB1, AMPK, mTORC1, and IGF-1 insulin. To explain it simply, ERK stands for extracellular signal-regulated kinase, AMPK for 5'-adenosine monophosphate activated protein kinase, mTORC1 for mammalian target of rapamycin complex 1, and IGF-1 for insulin-like growth factor 1. These pathways are collectively perturbed in old age to activate hallmarks of aging, which are mitochondrial dysfunction, inflammation, and cellular senescence. In aged organisms, the AMPK-mTORC1 axis is uniquely important for metabolic health, with notable effects in adipose tissue, and therapeutic inhibition of mTOR extends lifespan in mice. Aging studies to date have focused largely on lifespan extension, particularly in yeast, worms, and fruit flies, but lifespan extension does not necessarily reflect longer health span. There is a need for integrated studies to determine the effects of interventions on both health span and lifespan. Laboratory mice are particularly suited for such experiments, as aging pathologies that are important for human well-being and function are apparent in lifespan studies are well established in mice. The importance of chronic sterile inflammation for aging pathologies is increasingly recognized, and inflammation itself is a central hallmark of aging. In simplified terms, aging is associated with a dysfunctional adaptive immune system that is characterized by immunosenescence and thymic involution, along with inappropriate activation of innate immune genes such as IL-6, Interleukin 6. The pro-inflammatory signaling factors NF-kappa B, nuclear factor kappa light chain enhancer of activated B cell, and JAK-STAT3, Janus kinase signal transducer and activator of transcription protein 3, are specifically implicated in aging, and JAK inhibitors can alleviate age-related dysfunction. We proposed that IL-11, interleukin-11, a pro-inflammatory and pro-fibrotic member of the IL-6 family, may promote age-associated pathologies and reduce lifespan. This premise was founded on studies showing that IL-11 can activate ERK-mTORC1 and/or JAK-STAT3, the observation that IL-11 is upregulated in older people, and the fact that IL-11 is increasingly recognized to have a role in senescence, a hallmark of aging. Here, using a range of genetic and pharmacological approaches, we tested the hypothesis that IL-11 signaling has a negative effect on health span and lifespan in mice. IL-11 is upregulated with age. We determined IL-11 expression in the liver, visceral gonadal white adipose tissue (VWAD), and skeletal muscle gastrocnemius in mice over a time course of aging, which revealed progressive upregulation of IL-11 in all tissues. With age, there was progressive activation of ERK-P90RSK, inactivation of LKB1-AMPK and mTORP70S6K activation, which comprise an IL-11 signaling module, in liver and muscle. IL-11 upregulation was confirmed in livers, VWAT, and skeletal muscle of 2-year-old male and female mice. To identify the cell types that express IL-11 in old mice, we queried the tabula muris senis, which was uninformative, likely as IL-11 is expressed at low levels and IL-11 is largely translationally regulated. To better identify IL-11 expressing cells in tissues from old mice, we performed immunohistochemistry in tissues from aged IL-11 EGFP reporter mice. In 2-year-old IL-11 EGFP mice, IL-11 was apparent in parenchymal cells, hepatocytes in liver, adipocytes in VWAT and myocytes in skeletal muscle, and also seen in stromal, epithelial and endothelial cells across tissues and in nerves and skeletal muscle. Thus, IL-11 is expressed in diverse cell types in different tissues with aging. IL-11 is associated with senescence. To explore further the relationship between IL-11 upregulation and ERK-mTOR activation in tissues of old mice, we studied 10-week-old and 2-year-old IL-11RA1-, mice and wild-type littermate controls. IL-11RA1-, is typically a shorthand notation used in genetics and molecular biology. Let's break it down. IL-11RA1 is the name of a gene. Caret, --, indicates a specific genetic variation or mutation, which generally signifies a knockout or null allele. This means that both copies of the gene, one from each parent, have been inactivated or deleted. IL-11RA1 caret, --, would be read as IL-11RA1 knockout or IL-11RA1 null allele. on immunoblots of liver, VW80, and gastrocnemius extracts, old wild-type mice exhibited increased phosphorylated P, ERK and PP90RSK, increased PLKB1 leading to reduced PMPK, and increased levels of PMTOR, PP70S6K, and PS6RP. Levels of the canonical senescence markers P16-INCA4 and P21-WAF1/CIP1 were increased in tissues of old wild-type mice. By contrast, the phosphorylation status of these various kinases in S6RP and levels of P16 and P21 were similar between old IL-11RA1 knockout mice and young wild-type mice. So, there you have it. We've explored the complex interplay of aging, inflammation, and specific molecular pathways. Our research suggests that targeting IL-11 could potentially be a game-changer in combating age-related diseases. While significant strides have been done in understanding these processes, there's still much to uncover. Are you excited to continue exploring these pathways and their implications for human health and longevity? Thank you for watching. If you found this video informative, please like, share, leave comments, and subscribe for more updates on our research. And remember, healthy aging is a journey, not a destination. Let's explore it together. Next video will cover improvement of metabolism due to deletion of IL-11RA1 gene, IL-11 induced senescence in human, and deletion of IL-11 in female mice.